Hey everyone, we're going to take a look at Chapter 15, and Chapter 15 is going to cover the third line of the immune system, which is often referred to as the specific or the adaptive immune response. So let's go ahead and jump in with a slight recap from the last chapter. So to refresh your memory here, recall that our what we call the levels of defense, the three levels of defense, consist of barriers to infection, which really isn't technically part of our immune system, just keeping things out. Largely has to do with our anatomy and physiology. And then the second two, or excuse me, the second and the third, relate to the immune system itself. This is actually our immunity, hence the terms immunity here at the end. But the second is the non-specific, often involves kind of a general response to self versus non-self. And in the third line now, we're looking at how the what's called the specific or the adaptive line of the immune system works. Okay, so we're looking at kind of the last level of defense, arguably the most important level of defense. So I mean, they're all important, but without this last line, you tend to be unable to fight off any kind of infection. And there are certain diseases that are rare. There's certain rare diseases characterized by a loss or lack of the third line of the immune system. And typically these are very, very crippling diseases. Leaves people unable to fight off infection to a large degree. Now, one of the things I want to add real quickly to this diagram is the two key aspects of the third line. So the two, and this is a question I want you to know, the two key aspects of the third line are specificity and memory. Okay, so when we talk about the specific context of this third line, then that's obviously referencing specificity. So specificity, right? That seems to make sense. Now the other part, though, the adaptive component, this refers to the memory of our cells. It's not quite like memory of our brain, but memory in terms of the fact that our immune system can recall things that have been previously things we've been previously exposed to, and allow our body to fight them off in a more effective way upon repeated exposure. So the best way to kind of summarize this is enhanced immunity upon repeated exposure. As an example of this, and we'll talk about more specific examples towards the end of this chapter, but to give you an example of something you're kind of already familiar with, this is how a vaccine works. The vaccine is specifically designed to trigger the memory component of your immune system. What we do with the vaccine is we inject what looks like a live microbe, a live virus, sometimes a live bacteria, but it's actually not alive. It's considered inactive, weakened, or dead, depending on the exact vaccine and the exact context. So it's either killed or it's weakened to the point that it can't cause infection. However, our body recognizes it as if it was alive, or if it was at full capacity, and our body responds to it regardless. Now, when it does that, it creates memory cells, and those memory cells will stick around in our body for usually a number of years, up until the point where we might be exposed again. So if we're actually exposed for the first time, So if we're actually exposed to the pathogen at some point later, our body has created memory cells that recognize it. And what will happen then is they will respond to the pathogen. At that point, now the actual live pathogen, they will respond to it in an enhanced way because they've already seen it. They've already had an exposure in the past, and that enhances the immune response. It makes it more fast, more rapid, and more effective. So memory really is a way the body enhances a response after repeated exposure. So that's a very key part. This is why we don't tend to get sick from the same things over and over, and again, how a vaccine works. Now, the next thing I want to talk about here is the specificity of our immune system. When we talk about specificity, we're once again talking in terms that relate back to the lock and key analogy. So if you remember from some of the previous chapters, we've used the analogy of lock and key specificity. And the way this applies here is sorry, I keep doing that. It pertains to the cells that we call lymphocytes, B and T lymphocytes, and how they interact with what we call antigens or epitopes. And I'll explain the difference here in just a second. But essentially, our B and T cells are a cell. So we'll draw them. So a generic cell could be either a B or T cell. Later we'll clarify some of the differences between them, but for now we'll kind of talk about them as if they're the same thing. They're very similar. And what we find is on the surface of every B and T cell, we have these receptors. Okay. The receptors are the equivalent of the lock. So this is like the quote-unquote lock. Now the way it works then is that we have these substances called antigens. And antigens are the quote-unquote key. And an antigen, often a small substance, will actually bind to the tips of the receptors on the B and T cell. Now oftentimes they can actually bind to both ends of the receptor. So in this case it's kind of like a double lock where you can actually unlock it kind of from either direction, or from either side, excuse me. So if we blow this up just to make it look bigger, if we were to zoom in on this here, what we would find is that each tip of the Y, of that Y-shaped structure there, has its own little groove in it. So there's one side of it here, here's the other side of it over here. So each side almost has its own little locking mechanism, and then you'd have this connecting down here to the rest of the cell. So this is kind of like showing this tilted upwards. So what really happens here is the antigen has a shape that allows it to fit into the groove here, and you can actually have two antigens that can bind to each side of that receptor. Okay, so on the tips here there's grooves, and that's what allows the antigen to bind. Okay, but so similar to lock and key, not a perfect analogy, but this is how this works here. Okay, so the specificity goes beyond basic lock and key, and we find that our body has literally billions upon billions of lymphocytes, and each one has a specificity for a different antigen. So imagine a building with a billion doors to it and a billion potential locks that could fit any of those random doors, and that's kind of like your immune system. The doors in this case again being the B and T cells, antigens being a billion potential different keys to go into those locks. And so the way it works is we have different cells to bind to different antigens. Okay, we'll come back to this a little bit more here. So in other words, lock and key is in a very, very broad, wide-ranging context. Billions of locks, billions of keys, each one a little different. So what an antigen is it's really just any substance that our body can perceive as foreign by binding with these B and T cells. Okay, now usually they're some kind of protein and or some kind of polysaccharide, which is a large carbohydrate complex. They're not always, but they tend to be one of these two types of substances. So not necessarily everything can be an antigen. There also has to be a certain size component. They tend to have certain densities and things like that. But proteins come in many shapes and sizes, as do polysaccharides, so these are still a wide range of molecules. But each one is a little different, and it fits into the B and T receptor. Now the antigen is actually, or in many cases I should say, is actually composed of specific regions called the epitope. Okay, and if the antigen is the key, The epitope is like the specific grooves of the key that actually fit into the lock. So if this is a key here, to make this analogy a little more clear, this whole thing we could call the antigen. Okay, going again, sticking with the lock and key analogy here. This whole structure here, that's the antigen. Now, an epitope is the specific part of the antigen that actually fits into the B or T cell receptor. So in our analogy here, this specific portion will be what we call the epitope. Now this doesn't fit perfectly with the lock and key analogy, but I'm trying to give you the best idea of the difference here. So the antigen is like the whole thing, but oftentimes it's only one little part of it that actually interacts specifically with the B and T cell. Just like a key, you don't put the whole key into the lock, you put just the grooves on the end of it. So that's kind of like the difference between the antigen and the epitope. Here's another visual to give you a better idea of how that works. So here you're seeing the receptors. Sometimes we call these antibodies. I'll explain the difference later. An antibody is a type of B cell receptor, often one that is secreted from the cell. But anyway, it's really the same thing as a receptor. So here you're seeing in yellow the antigen, and then the epitope is what fits into the receptor there. Okay. It is possible for one antigen to have multiple epitopes. Here's a quick video showing you a visual on that, a better visual on that.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
So what they're suggesting here, what they're saying, is that something like a microbe, take an E. coli bacteria for example, a microbe will have many thousands potentially of different antigens and epitopes on their surface. So they don't just consist of one characterizing feature that's foreign, they consist of many. So for example, here's a microbe, like one that we've talked about, say for example E. coli. E. coli can have many different ways that the body will recognize it as being foreign. So it might have a structure here, and this would be one particular antigen epitope. It may have another structure over here, call that another antigen with its own epitope. Maybe have a slightly different structure over here, call that another one. And again, all throughout the surface of this cell, we can have literally hundreds, if not thousands, of slightly different features that all can be recognized as a different antigen and a different epitope. So when we talk about specificity and the ability of our body to respond to specific types of foreign substances, we're really talking about key regions, or I should say even subregions of a particular microbe. I should say pertains to subregions of a particular microbe. So our body could literally have a different lymphocyte to respond to each of these different parts of the cell. So our body could literally create 1,000 different types of lymphocytes, each one to respond to a different part of the microbe. Doesn't mean that it necessarily will. Sometimes the more you're exposed, the more different triggers that you'll have, the more different cells will respond to it. But the specificity really comes down to these key types of interactions. So over time, the more we're exposed to something, the more different types of lymphocytes we will trigger in response to a particular microbe. And we'll talk more about how exactly these types of cells come about and what are the ways that they can be triggered. So you wouldn't necessarily trigger all of these right off the bat, but through repeated exposure, as I mentioned, your immune system becomes enhanced. So as you become exposed more and more, you would start to potentially anyway trigger more of these lymphocytes. Now theoretically it only takes one to kill the cell. So theoretically, by just simply triggering one type of lymphocyte against it, you would theoretically be able to kill it. But the more the better, and the more effective your immune system will become. So hopefully this is making sense so far here. So specificity goes back to lock and key, and it really pertains to the key subregions of a microbe referred to as the antigen and the epitopes, of which a single microbe could literally have hundreds, if not thousands. Now, memory is the other key aspect, and memory relates to the way the body responds to things over a long-term basis, and how, as I already mentioned, by which repeated exposure increases and enhances our immune function. So let me add a little bit more to this here. When we are first exposed, prior to being first exposed, we back up here, we have what are often referred to as naive B and T cells. And generally these are cells that have yet to be exposed to any sort of foreign antigen. So in other words, they've never actually had any sort of encounter with a foreign substance. Okay, so what happens with these cells is they start out in this naive state. Now, we'll talk more about how they come to be shortly. So for now, we're just calling them naive, meaning no prior exposure to matching antigen. Okay, so what that means is, again, they've never, the particular antigen that matches that cell and the receptors, the specific lock and key interaction, has never actually taken place. Okay, so these cells are actually created and then they wait before they're ever exposed to an antigen. Okay, that'll make a little more sense here later. So they start out as naive. When the antigen finally does bind, if it ever does, sometimes it won't, but if and when the antigen does bind and we have that lock and key interaction, what that does is it actually triggers a response by these cells, and the naive B or T cell will actually differentiate and proliferate into two subtypes of cells, two general subtypes. Okay, and one subtype are referred to as memory cells. The other subtype can be generally referred to as effector cells. The general difference is that effector cells will have an immediate effect. Immediate effect against antigen / microbe, whatever it is they're responding to. So in other words, when you're exposed, theoretically there's a problem, and the naive cells differentiate partly into cells that are going to handle the immediate problem. Now, because we may be exposed to that microbe or antigen later, and these cells tend to only last a short period of time, our bodies have evolved to create a long-term solution to the problem, which are called memory cells. What memory cells will do is they will persist in the body long after infection, or say long after exposure to antigen / microbe. And further, they will have a rapid effect, or I should say a enhanced effect, enhanced response, let's say, to secondary / or let's just say enhanced response to next exposure, whatever that may be. So let's say you're exposed to the same microbe a couple weeks later, or maybe 2 years later. These cells will still be around in your body, and when your body encounters the antigen again, what will actually happen, let's say here's the antigen over here again, you've been exposed to it for a second time. These cells will also have receptors, same receptor here. So when the antigen binds to it, what will actually happen is these cells will transition back into effector cells, and they will go after the antigen and attack it. So memory cells will basically jump right back into the effector cells if you're exposed again, and then you'll create more memory cells, and that process will repeat. So you start with naive cells, they differentiate into memory and effector cells. Effector cells go on to, quote unquote, kill/eliminate, whereas the memory cells are kind of hanging back. They're just kind of waiting to see what's going to happen long term. Microbe comes back, antigen comes back, memory cells go back into effector cells, which are going to kill and eliminate. And they'll create more memory cells, so the process will repeat over and over and over again. So that's kind of how memory works, and this is why we don't tend to get sick over the same things over and over and over. And this is why our body tends to get more effective as we get older. So if you think about this, little kids tend to get sick all the time, partly because they're, at a certain age anyway, they tend to put stuff in their mouth, which is kind of a natural way to build immunity. Also how they kind of sense the world. But part of that has to do with their immune system as well, a big part of that. The immune system takes years to kind of build up an effective immune response and effective memory cell. When you're first developing at a young age, more or less everything you're exposed to is you're being exposed to for the first time. Not quite literally, but figuratively speaking, you're kind of having a first exposure over and over and over. You don't really have that many memory cells, and so your body's constantly being challenged, and that's why little kids tend to get sick more often. Also, the immune system doesn't really fully develop until really around puberty is when it fully develops, but especially before the age of 5. Around the age of 5, your gut immunity becomes much more effective, and so you tend to hit a much more effective range around the age of 5. So prior to that, you really don't have all the cells fully developed yet. So it's not just about the memory cells, it's also about just the overall construct of your immune system. Our body really, when we're first born, is still in rapid development. I mean, obviously, physiologically or structurally, it's fairly obvious, size, and our brain tissue hasn't fully developed, but it's also true of the immune system. Just like your bones haven't reached full capacity when you're 3 years old, neither has your immune system. It's still growing as well. So there's a lot of reasons why, but memory cells are a part of that. Now, what I mentioned earlier is that the third line of the immune system is absolutely critical to the overall effectiveness of your levels of defense. The first level and the second level are necessary and important. The first level is keeping things out. The second level is responding to things that get past. But ultimately what we'll find is that primary pathogens, which we know exist, primary pathogens will be able to at some point get past these first two levels. You'll be exposed to a virus that gets into the respiratory tract. The second line of defense won't be able to handle it effectively. You'll be exposed to a bacteria in a sufficient effective dose. You may eat some bad food. It gets into your gut. Phagocytes, other innate factors aren't able to eliminate it. You're going to eventually have to have a third line to handle these things that don't get dealt with effectively by the first two lines. And really that's when we see the full importance of the third line. And to clarify here, immunocompetence really is a factor of this third line of the defense here. So individuals that are considered to be immune compromised, a term I've used before, we talked about opportunistic infections and how they tend to infect immunocompromised people. Immune compromised individuals tend to lack a third line of defense, and they don't have either enough of a memory or B and T cell population, I should say. There may be problems with the bone marrow. There could be cancer and chemotherapy, which is suppressing bone marrow, which is where these cells originate, as we'll talk about shortly here. It could be that they have a virus like HIV, or it could be that their body is just in such a bad physiological condition it's not able to function properly. So such as what you see with like type 2 diabetes that's unmanaged and unproperly dealt with. So ultimately, the ability to be healthy and to have a functional immune system ultimately relies on the third line of the immune system. It's just a matter of time before you need it, and without it, you're kind of, I wouldn't say kind of, you're definitely vulnerable to infection. Okay, so that's kind of what that is in reference to there, to clarify what we talked about previously.